Hello to everyone here in Lipset and to everyone joining online. It's my distinct pleasure to introduce Dr. Lei Wang today. One quick note before we get started. The CME code is 57654. If you have a question during the seminar or after the seminar, these can be submitted via the Submit Feedback button online. And for those here in person, there'll be a reception that follows the lecture in the hallway outside the seminar hall. So currently, Professor Wang is a professor in the Department of Pharmaceutical Chemistry at UCSF, where he's been since 2014. He performed his bachelor's and master's studies at Peking University before moving to his PhD studies in the lab of Peter Schultz, where he helped to pioneer methods to expand the genetic code for translational reprogramming and incorporation of non-natural amino acids. He then performed his postdoc studies with Roger Tsien at UCSD to develop new fluorescent protein sensors via mutagenesis-based methods. He began his independent career as an assistant professor at the Salk Institute in 2005, where he stayed until 2014 when he joined UCSF. Dr. Wang has received several awards, including the Emil Thomas Kaiser Award from the Protein Society for significant contributions to the study of proteins, as well as an NIH Director's Innovation Award, amongst many others. As Dr. Wang will tell you today, his lab develops new methods based on genetic incorporation of non-natural amino acids to introduce novel functionality into proteins, to investigate biological processes in vivo, which he'll cover during his talk today. So join me in welcoming Dr. Wang.
Thank you very much, Ross, for the kind invitation and generous introduction. I had a great time meeting with the outstanding PIs and the trainees here. Really enjoyed the visit. It's a great honor and a pleasure to be here. My talk today will be biospecific chemistry for covalent linking of biomacromolecules. Before we proceed, I'd like to disclose a potential conflict of interest. I serve as a scientific advisor to another therapeutics biotech company focusing on developing covalent biologics. Just test my cursor, as you can see. Okay, great. In nearly all life forms, proteins interacting with proteins and other classes of biomolecules to perform the essential biological functions. These interactions, including electrostatic forces, van der Waals forces, pi effect, and hydrophobic interactions, are primarily non-covalent in nature. As a result, they tend to be weak, transient, and reversible. Our goal is trying to engineer such non-covalent interactions into covalent bonds, creating strong, stable, and irreversible connections for biomacromolecules to study and harness biological activities. Proteins have a limited ability to form covalent bonds. A key example is the disulfide bond formed between two cysteine residues spontaneously. While crucial for protein structure and function, the disulfide bond is reversible and sensitive to redox conditions. Another major mechanism is through catalysis, such as the formation of the isopeptide bond between the lysine residue and an aspartic residue, or the attachment of ubiquitin to other proteins through the enzyme-mediated catalysis. As you can see, this will require multiple residues and a special microenvironment. And however, rationally designing these bonds remains challenging due to the complexity of these processes. Bioorthogonal chemistry has revolutionized the covalent modification of biomolecules. In this approach, two complementary bioorthogonal functional groups are introduced separately into a small molecule and a biomolecule. which then click to form the connection. Because the targeted biomolecule must be modified with a bioorthogonal functional group, implementing this approach in vivo is challenging. In particular, especially for therapeutic applications where the targeted molecule are very difficult to be altered. To address this, we propose biospecific chemistry. This approach introduced a single bioreactive group into a biomolecule, which then react with another biomolecule without the need to modify the latter. This ability to covalently modify native biomolecules should make biospecific chemistry ideal for in vivo studies and therapeutic applications. We began exploring biospecific chemistry to enable proteins to covalent target another protein. Beyond the disulfide bound, we envisioned whether additional covalent linkages could form spontaneously between an unnatural amino acid and a natural amino acid residue. This, we call it bioreactive unnatural amino acids, if successful, would break the natural barrier, introducing new covalent bonding capabilities into proteins. Unnatural amino acids can be site-specifically introduced into proteins through genetic code expansion, using an orthogonal tRNA synthetase pair to suppress an amber stop codon artificially introduced into the RNA. However, the incorporation of a bioreactive unnatural amino acid into proteins in cells has long been considered infeasible. For a bioreactive unnatural amino acid to function, it must react with biomolecules inside the cells. But such reaction risks cytotoxicity and could store translation if it reacts with the translation machinery. Additionally, the abundance of natural amino acids in proteins and in cells pose a challenge for non-selective reactions, which can compromise specificity. To address these challenges, we developed a strategy called proximity-enabled bioreactivity. In this approach, the UAA, now I refer unnatural amino acids as UAA, the UAA's reactivity is designed to be latent inside the cells, preventing unwanted reactions within intracellular molecules and allowing for genetic incorporation. Once incorporated into protein and brought close to the targeted natural amino acid residue, the UAA's reactivity is then triggered by the proximity effect, which reduces the entropy of activation and increases the effective concentration, enabling a covalent bond to form selectively between the unnatural amino acid and the targeted residue. The reactions we aim to develop must be both biocompatible and efficient under physiological conditions, which is a challenging balance to achieve. We focused initially on targeting cysteine residue due to its high nuclear philicity. Our hypothesis was that a weak electrophile could be stay inert in cells, but selectively react with cysteine when brought in close proximity. To test this, we choose fluoromethyl ketone as the warhead and designed the unnatural amino acid we called F-FACT. We incorporate F-FACT into a model protein, into an antibody, and express this in E. coli. After purification, mass spec verified incorporation of this unnatural amino acid, and we don't see any additional modification to the F-FACT, including unintended reactions with intracellular cysteine or glutathione. This results indicating that F-FACT stays inactive during the protein biosynthesis and purification. To test whether F effect can form a covalent bond with the cysteine in proteins, we incorporate the F effect into an antibody and place the cysteine residue into the Z protein at their binding interface. And these two proteins were purified and incubated. And we then run an SDS page under denaturing conditions to test whether these two react with each other. The results shows a covalent complex of antibody Z was formed. However, if we replace the F effect with the non-reactive isosteric F act, or place the cysteine with other amino acid residues, or put the cysteine into different positions, no covalent complex formed. So this results indicating that the covalent bond formation between these two proteins depends on the proximity of F effect and cysteine. Next, we tested the intramolecular reactivity in the red fluorescent protein M-plan. We replaced the tyrosine 67 of the fluorophore with the unnatural amino acid F effect, and replaced the serine 146 on the beta barrel with cysteine. We then expressed this mutant protein in E. coli, purified it, and mass spec confirmed nearly 100% efficiency in the covalent bond formation. And single molecule analysis of this mutant protein showed that this single covalent bond doubled the lifespan and the photon output of this mutant fluorescent protein. These are benefits that are very difficult to achieve using only natural amino acids. Building on this breakthrough, we and several other research groups have since designed and genetically incorporated a variety of latent bioreactive unnatural amino acids to target different amino acid residues. Here are some representative examples. As you can see, besides cysteine, we are now able to covalent target several different other natural amino acid residues through this proximity-enabled reactivity mechanism, highlighting the generality of this approach. One versatile UAA we have developed is FSY for its exceptional biocompatibility and multi-targeted ability within live cells. Inspired by the pioneering work of Barry Sharpless on click chemistry, we designed this FSY to contain a fluorosulfonate functional group. We incorporated FSY at the binding interface of antibody Z and introduced different nucleophilic amino acid residues to test whether FSY can react with them through the sulfex chemistry. And we express these two proteins directly in E. coli without purifying them directly in live cells. We then analyze the cell lysate. As you can see here, both on the Western blot and SDS page, the results showed that the FSY was able to react with lysine, histidine, and tyrosine. And this reaction specificity was further verified by mass spec. Since lysine, histidine, and tyrosine are abundant at many protein interaction interfaces, so FSY has the potential to target virtually any protein. Another versatile reaction is PFAX, the phosphorus fluoride exchange reaction. While PFAX typically requires a catalyst for reactions between small molecules, we discovered through genetic incorporation of these two unnatural amino acids into proteins, we found in the protein environment the PFAX reaction can now be activated simply by the proximity effect. And this allows we use PFAX to target tyrosine, histidine, lysine, and cysteine residues directly in live cells. Additionally, the PFAX reaction is really interesting. When it reacts with histidine, it actually displays very interesting thermal sensitivity as well. What can we achieve with this new covalent bonding abilities? First, I would like to introduce genetically encoded chemical cross-linking, or JAKs, to identify protein-protein interactions. One application we developed is trying to use this to probe ligand-receptor interaction on the live mammalian cell surface. When we experience stress, the hypothalamus will release corticotropin release factor, CRF, a peptide hormone that binds to a class B GPCR, CRF1R or CRF1BR, triggering a signaling cascade that stimulates our heart, our brain, and muscle, resulting in stress response. However, chronic stress can lead to anxiety and depression. And blocking the CRF receptor interaction could potentially treat these conditions. But at the time, it was unclear how the CRF peptide hormone binds and activates the receptor. Then, as an assistant professor, under the pressure of convincing NIH study panels seeking promotion and publishing, I was immediately intrigued by the possibility of creating a happily ever after pill by blocking this interaction. So my group started to mapping ligand binding sites on the GPCR using the photo cross-linking UAA. We used the unnatural amino acid, we call it AZI. We can incorporate this AZI into various different positions into this GPCR, which was expressed directly in live mammalian cells. And then we throw in the peptide hormone, perform the photo cross-linking directly on live mammalian cell surface. After which, we just analyze the cell lysate to detect whether the ligand is cross-linked with the GPCR. As you can see in this panel A, when we incorporate AZI into specific size of this GPCR, it can capture the ligand. We can see a covalent complex of ligand detected here. And this size are potentially the ligand binding sites and marked in magenta here on the GPCR transmembrane domain structure. So we were very happy. But at that time, we realized this still cannot help us to correctly positioning the CRF ligand into the binding pocket of this GPCR, because photo cross-linking is not residue selective. So we cannot know the distance constraints between the peptide ligand and the GPCR receptor. And here, JAX provided the solution. Since we now know JAX is both residue and distance specific, we could use it to pinpoint the exact contact points between the CRF and the receptor. So what we did is introduce this F-effect unnatural amino acid into different positions of this CRF1R and assisting into the peptide ligand. We throw in this peptide ligand into the live cells and then analyze if the receptor captured the ligand using Western blot analysis of the cell lysate. And this result tells us which residue in the receptor is in contact with a specific residue in the peptide ligand, as shown by the matching color in this diagram here. So based on this constraints information, as well as the structure of individual domains of GPCR separately, now we were able to build a model for the CRF ligand binding to the full-length GPCR, providing mechanistic insights into receptor activation and inhibition. As you can see here, all the information we obtained is directly on the live mammalian cell surface, which can complement what people can obtain later using the crystallography or cryo-EM structures. Taking a bold step beyond the cell surface, we next applied JAX inside the live cells to capture protein-protein interactions. So the strategy is to incorporate the latent unnatural amino acid into our target protein, which is expressed within live cells. Whenever an unknown protein interacts with our protein of interest, then the bioreactive unnatural amino acid will react with a natural residue onto this unknown protein, irreversibly capture it. This will allow us to do subsequent processing and then identify this unknown protein using mass spec. So as you can see, in addition to working in live cells, our JAX mechanism has a major advantage is its spontaneous reaction and a long reaction window. which allow it to enrich weak and transient protein interactions, improving the sensitivity for identification. Thyroidoxin is an enzyme that can reduce the disulfide bound of proteins. To identify thyroidoxin's substrate protein in E. coli, we incorporate this UAA BPRY into near the active site of thyroidoxin, positioning the side chain toward the cysteine residue of the substrate protein. We simply express this BPRY containing thyroidoxin in E. coli, and Western blot analysis of the cell lysate showed multiple bands with higher molecular weight containing thyroidoxin. And these bands were then pulled down and identified by the mass spectrometry. This gives us 205 proteins that directly binds to thyroidoxin. And the resulting protein interaction network provides new insights into thyroidoxin's role in regulating energy metabolism, DNA damage repair, and other processes. Of course, cysteine is not always available for targeting, and that's why we developed FSY and FSK, these two unnatural amino acids, to target the more abundant tyrosine, lysine, and histidine residue, which should broaden the range of proteins we can capture. Additionally, we think FSY and FSK can be placed into the periphery of the protein instead of the binding interface or the active site to minimize interference. So to test this idea, we incorporate FSY and FSK again into thyroidoxin, but this time further away from the active site. We then express these mutant proteins in E. coli, and now you can see on Western blot, we can identify even more substrate proteins of thyroidoxin compared to using the BPRY at the active site. In addition to covalently targeting proteins, we recently enabled genetically encoded chemical cross-linking of RNA and other different classes of biomolecules. Protein-RNA interactions regulate nearly all aspects of RNA, including pre-RNA splicing, RNA modification, translation, and degradation. Recent studies have also revealed that RNA binding proteins can even bind RNA through the very intrinsic disordered regions. So to fully understand this complex regulatory mechanism and uncover new insights, it is crucial to identify protein-RNA interactions in vivo, ideally with single nucleotide and a single amino acid resolution. The most widely used method currently is the nucleoside-based UV cross-linking. When exposed to UV light, the base on the nucleoside can generate radicals, which can cross-link nearby amino acid residues. Combined with immunoprecipitation and high-throughput sequencing, we can identify the RNA targets of specific RNA binding proteins. Unfortunately, UV cross-linking has a bias preference for the uridine, therefore making it difficult to study RNA regions that are lacking uridine. In addition, UV cross-linking, as I mentioned earlier, is non-residue selective, and therefore making it challenging to achieve amino acid resolution. And because the reaction is non-specific, therefore it's infeasible to engineer precise covalent linkages between proteins and RNA. Our JAX-RNA strategy is to incorporate a latent unnatural amino acid into the RNA binding protein. And this UAA will react with a nearby nucleotide on the RNA when the RNA binding protein interacts with the RNA. As you can see, now the reactivity comes from the protein instead of coming from the RNA. If we are able to target a 2' hydroxyl group on the ribose, which is present in all RNA nucleotides, we should be able to eliminate the nucleotide bias. And since proximity enables reactivity, doesn't require an external trigger, so we think this JAX RNA should work both in live cells and in vivo. In addition, the reaction occurs exclusively between the unnatural amino acid and the nearby nucleotide, and therefore should provide a single amino acid resolution and a single nucleotide resolution. We use the Cas13b as our model system, which cleaves precursor guide RNA into mature guide RNA. On the basis of the crystal structure of Cas13b, we incorporated FSY into the near the cleavage site, positioning FSY's side chain toward the 2' hydroxyl group of the bound RNA. After incubating this mutant Cas13b with precursor guide RNA, we found that it was able to capture the precursor guide RNA regardless of the nucleotide identity at the cleavage site. This demonstrates that JAX RNA can indeed covalently target RNA without the nucleotide bias. To demonstrate the utility of JAX RNA, we developed a grip-seq technology to map M6A modification across the RNA transcriptome. M6A is a widespread RNA modification that regulates mRNA function. While several detection methods already exist, they either lack single nucleotide resolution or lack protein specificity, or they rely on the antibody for in vitro recognition. Our goal is trying to detect the M6A directly in cells with both single nucleotide resolution and protein specificity. Our strategy involves expressing the M6A reader protein in live cells to recognize M6A. And latent bioreactive unnatural amino acid is incorporated into the reader protein near the M6A binding site. Once the reader protein recognizes M6A through the JAX RNA mechanism, the UAA then will cross-link a nearby nucleotide on this RNA. And this covalent complex is then pulled down, and then the protein was digested to release the captured RNA. This captured RNA is then reverse transcribed with the process terminated at the cross-linking site due to the presence of the unnatural amino acid. And this RNA transcripts will be PCR amplified and then sequenced in high throughput. And this will reveal all the cross-linking sites. Then, once we know the cross-linking sites, we then expect the M6A site should be adjacent to the identified cross-linking sites. So for this experiment, we used the YTH domain of the human M6A reader protein, YTH-DF1, as the M6A reader protein. And we incorporated unnatural amino acid SFY into the M6A binding site of this YTH domain and putting it close to the M6A shown in blue here. We tested this grip-seq technology in HEC293 T cells. And here shows the high throughput sequencing peaks of one example. As you can see, the peaks terminated already, some of them at the M6A site. We then analyzed all the nucleotides surrounding these termination peaks and found they are enriched with this dark motif, which is consistent with the consensus motif of M6A. In addition, we found that the cross-linking site are primarily at the -3rd position relative to the M6A modification, indicating this approach gives us a single nucleotide resolution. So using this approach, we predicted a total of almost 14,000 M6A sites using this YTH domain, and among which about 6,000 is a known M6A sites verifying over this approach, and the rest, almost 8,000, is newly identified. In summary, as you can see, our method can recognize M6A directly in live cells, has a reader protein specificity, and therefore we anticipate this can be similarly be used to map all the RNA modifications in cells, as long as you have a protein to recognize the RNA modification. Next, I would like to discuss genetically encoded chemical cross-linking of carbohydrates, a method we refer to as Jack's sugar. Protein-glycan interactions play a crucial role in various biological processes, ranging from interspecies communication to interspecies recognition. For instance, the binding of Siglec protein to the sialoglycan can inhibit immune activation, and which is actually the self-recognition in our immune system. And bacteria and viral pathogens can exploit sialoglycan mimics to inhibit Siglec on the immune cells, therefore to evade immune detection. Similarly, tumor cells can upregulate the sialoglycan on their surface and use this to engage the Siglec on the immune cells, leading to immune escape and cancer progression and metastasis. Despite their central role in molecular recognition, the protein-glycan interactions have been very difficult to study. Among many other difficulties, a major challenge I want to highlight here is the low affinity between a single sugar protein recognition, with KD dissociation constant often in the high micromolar and even in the millimolar range. Since the binding affinity is so low, so we thought, can we covalently stabilize protein-glycan interaction, therefore to better study it? However, we found that there was no reaction that can covalently react with glycan under very mild conditions. So we have to start to identify a functional group that can react with glycan under biocompatible conditions. To do this, we developed a plan, we call it plan and cast cross-linking strategy. We first incubate a Siglec protein with its ligand, sialoglycan ligand GD3. When these two bind with each other, then we throw in a bunch of cross-linkers. These cross-linkers have two ends. One end reacts rapidly with the protein, plant the cross-linker on the surface of the protein, and then cast the other end toward the bound glycan. If this end reacts with our glycan, then a covalent complex of glycan and protein will be formed, allow detection. So we designed and synthesized several small molecule cross-linkers and discovered that this functional group, aerosulfonyl fluoride, was able to react with glycan under mild conditions. As you can see here, this is the only positive spot we detected. Next, we then designed this unnatural amino acid SFY to contain the identified sulfonyl fluoride functional group. And we evolved the new tRNA synthase to incorporate this unnatural amino acid into the Siglec-7 binding protein. We found once this unnatural amino acid is incorporated into this 3 specific sites, it enabled the Siglec-7 to bind GD3 covalently. Next, we then incubated the cancer cell line that has high level of sialoglycan with Siglec-7 or covalent Siglec-7 SFY. As you can see, the covalent version of Siglec-7 has a dramatic increase of cell binding compared to the Y-type Siglec-7, as shown in this graph here. Furthermore, if we pre-treat the cells with sialidase to remove the cell surface sialoglycan and repeat the experiment, now you see the cell binding is dramatically reduced to 1/10 of the original level. This results in supporting that Siglec-7V SFY cross-linked with sialoglycan on this cancer cell surface. Many tumors upregulate the cell surface sialoglycan, and this sialoglycan can engage the Siglec-7 receptor on human natural killer cells. This sends an inhibitory signal to damp down the NK's ability to kill the cancer cells. We envisioned if we throw in an irreversible binder, the Siglec-7 VSFY, when this irreversibly bind to the cancer cell surface glycan, this will release the NK cell from the signaling, inhibitory signaling axis, and therefore enhance the NK cell's ability to kill cancer cell. To test this hypothesis, we treated 3 different cancer cell lines that has high level of sialoglycan with either Y-type Siglec-7 or our covalent version of Siglec-7 SFY. Then we wash the cells and then add back to the NK cells and measure the cancer cell death. As you can see here in these 3 graphs, shown in red is our covalent version of Siglec-7V. They significantly enhanced the NK cell's ability to kill cancer cells compared to the Y-type in black here. And these findings suggest that covalent targeting the glycan on the cancer cell surface might be a new approach for cancer immunotherapy. In the final part of my talk, I will discuss the development of covalent protein drugs using proximity-enabled reactive therapeutics, or PEREX for short. Covalent small molecules can form a covalent bond with their target after binding, and this covalent linkage offers several advantages over the non-covalent drugs, such as increased potency, prolonged duration of action, less frequent dosing, and the ability to target the previously undruggable proteins. Although the pharmaceutical industry historically avoided them due to the concern of off-target reaction, covalent small molecule drugs have resurged with great success in the recent years. In contrast, the potential of covalent protein drugs remains largely unexplored due to proteins typically lack the ability to form covalent bonds with their targets. To enable covalent protein drugs, we developed a platform technology we called proximity-enabled reactive therapeutics. In this approach, similarly, we incorporate a latent bioreactive unnatural amino acid into the protein drug near the binding interface. Once the protein drug binds with its target, then the unnatural amino acid will react with a nearby target residue, irreversibly cross-link the protein drug to its target, essentially serving as a covalent protein drug. During the COVID pandemic, we developed a covalent nanobodies trying to inhibit a SARS-CoV-2 infection using this PEREX mechanism. As we all know, this virus enters the cells first by binding its spike protein with the human ACE2 receptor. Conventional antibodies and nanobodies can bind to the spike to inhibit this process, but such binding is non-covalent and therefore is reversible. And this makes the complete inhibition quite challenging. We thought if we use a covalent nanobody to irreversibly bind to the spike protein of the virus, this may reduce the likelihood of viral escape and significantly improve to achieve more potent inhibition. We then worked with a nanobody MMB6 to find out a site that can do the cross-linking. We actually screened all 30 sites in the 3 CDR regions of this nanobody using FSY. As you can see in this cross-linking analysis, we found multiple sites enabling the cross-linking of the viral spike protein in CDR1 region here. In CDR3 region, we found multiple sites that can enable efficient cross-linking. Because this virus infection is a very acute process, we would like to accelerate the reaction. To do this, we introduced a fluorine electron withdrawing group onto the FSY, creating the new unnatural amino acid FFY. And this increased the reaction rate by 2.4-fold. In the best cases, when we incorporate FFY at site 108 of this nanobody MMB6, it can cross-link the spike protein in vitro within 10 minutes. To evaluate the neutralization efficiency, we treated a pseudovirus of a SARS-CoV-2 with either a Y-type nanobody or our covalent nanobody. And then we used this mixture to infect the ACE2 expressing 293 T cells. The infected cells were then measured and plotted against the concentration of the nanobody added. As you can see in this plot, in gray is the Y-type nanobody, which has IC50 of 36 nanomolar. In contrast, our covalent nanobody reached the IC50 of 1 nanomolar, suggesting a 36-fold improvement. We further tested these nanobodies against the authentic SARS-CoV-2 virus. Again, as you can see here, the covalent nanobody achieved a 40-fold improvement compared to the Y-type nanobody. Representing, this result indicated that a significant increase in potency when you have a covalent bonding ability in neutralizing this Y-type SARS-CoV-2. In addition to the Y-type virus, MMB6 was also able to bind different variants, including alpha, delta, epsilon, and lambda. And correspondingly, when we tested the cross-linking, we found our covalent nanobody was able to cross-link this covalent variants as well. In addition, they also showed a drastic increase in the potency in neutralizing this SARS-CoV-2 variants. But the MMB6 nanobody was unable to bind the latest Omicron variant. So we then engineered another nanobody called MB70, which was able to bind the Omicron variant. And similarly, this covalent version of the MB70 cross-linked the Omicron variant in high efficiency. This actually can achieve significant cross-linking within 5 minutes. And as you can see here, the covalent nanobody also showed 8 to 10-fold increase improvement over the Y-type nanobody in neutralizing this Omicron variant. So in summary, we found that the covalent nanobody are significantly more potent than the Y-type counterparts in neutralizing both Y-type SARS-CoV-2 and its variants. In a second example, I'd like to demonstrate how PEREX can address a long-standing challenge in the targeted radionuclide therapy, the TRT. TRT basically aims to deliver radio activity into tumors while minimizing the exposure to normal tissue. And radiation has a very unique ability to kill cells regardless of the disease mechanism, and therefore is currently experiencing a clinical resurgence. However, a major challenge in the field has been how to balance the efficacy with safety. As you can see here, if you use conventional antibody to deliver the radioisotope, you can achieve a very effective tumor accumulation. But because antibodies can stay in the bloodstream for a long time, this increases the damage to the normal tissues. In contrast, if you use low molecular weight ligands to deliver the radioisotope, this can be cleared from our blood very quickly, providing a safer profile, but in the meantime also reduce tumor uptake and reduce the efficacy. An ideal radiopharmaceutical would be combining these two. What if we have a faster blood clearance and higher tumor uptake? That would be perfect. So we thought that a small covalent protein could serve as an ideal vehicle for radiopharmaceutical because its low molecular weight will allow it to rapidly be cleared from the bloodstream. But in the meantime, once it irreversibly binds to the cancer antigen, it can stay the tumor site for a longer time to achieve more effective therapeutics. So to test this concept, we began with a covalent nanobody specific to the HER2 receptor. Similarly, we incorporated the unnatural amino acid FSY into the nanobody at this D54 site to target the HER2 by targeting its lysine 150 site. And in vitro, we showed this covalent nanobody FSY could cross-link the HER2 extracellular domain very efficiently, and significant cross-linking could be detected within 10 minutes. We then showed that this nanobody was also able to cross-link the endogenous HER2 receptor on cell surface in different cancer cell lines. In these two cell lines, they lack HER2 expression, and therefore we didn't detect any cross-linking, suggesting this cross-linking is highly specific. We further showed that this NB nanobody FSY was able to cross-link the HER2 receptor on the tumor directly in mice as well. We next labeled these nanobodies with iodine-124 and inject them into mice xenografted with HER2-expressing tumors, followed by the PET imaging. To your right side, from 3 to 10 hours post-injection, as you can see, there was no significant difference between the Y-type nanobody, the top here, and the covalent nanobody in terms of a tumor signal. However, a dramatic difference can be found from 24 hours to 72 hours. While the nanobody, the Y-type nanobody was no longer detectable, the covalent nanobody remains clearly seen. Furthermore, if we do 3D imaging, as you can see here, from 24 to 72 hours, the tumor signal was distinctly visible using the covalent nanobody, but not visible in the Y-type nanobody. And another exceptional observation is that we see very clean background in all the normal tissues, except the thyroid, because it's the natural site for iodine metabolism. And the total radiation accumulated in the tumor, shown on left here, is 81% more with the covalent nanobody. So these results tell us that the covalent nanobody significantly prolonged the tumor retention as well as increased the tumor accumulation of the radioisotope. Well, would this be sufficient to impact the anti-tumor effect? So to test this, we then labeled the nanobody with actinium-225, a potent alpha emitter, and then inject this nanobody into the mice xenografted with HER2-expressing tumors. As you can see in this graph here, compared to the saline control in black, the Y-type labeled, the actinium-labeled Y-type nanobody in blue showed no significant difference in terms of tumor inhibition. In stark contrast, in orange here, the covalent nanobody labeled by the actinium-25 shows a significant tumor inhibition. And endpoint analysis of the tumor weight also indicate the covalent nanobody reduced the tumor weight significantly, but not with the Y-type nanobody. And the body weight of the mice did not change significantly across all three groups, suggesting there's no systematic toxicity. We further did a tissue analysis of the liver, the kidney, the heart, and the brain and the bone here, and we didn't find any abnormalities, suggesting there's no toxicities either here. So in summary, the radiopharmaceutical using this covalent nanobody as the delivery vehicle shows potential for more effective tumor inhibition without causing tissue toxicity. To conclude my talk, by developing latent bioreactive unnatural amino acids through the proximity-enabled bioreactive mechanism, we can now genetically introduce covalent linkages between proteins as well as between proteins and other biomolecules, including RNA and carbohydrates. These covalent connections are selective, stable, and irreversible, opening up new possibilities for creating novel protein properties, which I don't cover today. And we can also use these covalent linkages to study biomolecule interactions in cells and in vivo. Additionally, we developed this platform technology, Parex, for generating covalent protein drugs, which has shown promise in cancer immunotherapy, SARS-CoV-2 neutralization, and targeted radionuclide therapy. We anticipate that shifting the paradigm from non-covalent to covalent interactions in protein biomolecular dynamics could greatly advance synthetic biology, basic biological research, and biotherapeutics. Finally, I'd like to thank my group members, both past and present, for making all this possible. And I'm also deeply grateful for the generous support of many wonderful collaborators as listed here and the various funding organizations, with special thanks to NIH for your continuous support. Thank you very much for your attention. I'll be happy to take questions.
So there are mics on either side. Please use. No questions yet coming in online.
Hi, Lei. Really great talk. So I have sort of a conceptual level question. So you have different latent bioreactive amino acids, and they have sort of different levels of reactivity. Do you think that you push the reactivity as far as you can without encountering some sort of autoreactivity, or do you think that it will be possible to make amino acids that have higher rates of reactivity in the context of pole-sized proteins?
Yeah, that's a great question. We have a wide range of warheads with different reactivity than self. We actually studied this reaction using different protein systems. For example, they have different binding affinities, etc. We found the reaction depends on multiple factors. The intrinsic reactivity of the warhead is one aspect of it. The binding affinity is another part, and the orientation, the distance, everything included contributes to the final reactivity. To give you an example, some of the reactions may take multiple hours, 6 hours or 8 hours to achieve 80%. And some systems we found that this cross-linking can occur within 5 minutes. And in some systems we found that the cross-linking can reach 99% completion. Whereas many other systems, as you suggested, we do see intramolecular cross-link. And generally, as you suggested, this intramolecular cross-link is often correlated with the reactivity of the warhead. So I'm hoping that with the advancement of new chemistry, we may be able to find an even better warhead than what we currently have to minimize the intermolecular cross-linking while significantly accelerate the final reaction, for example, between two proteins instead of having been quenched by intermolecular reactions.
Great. Thank you.
Hi. This is an amazing, creative, and thought-provoking presentation. Congratulations. I'm just wondering about the path to commercialization, and we know how difficult it is here to put our small molecules onto a translational path. I imagine that if you propose to a company that you have some covalent warhead on your protein or on your small molecule, the path would be even harder. What can you say about the de-risking or potential toxicity of the warhead? You mentioned in one case you didn't have any reactivity with glutathione, but what evidence do you have that there is not other toxicity? For instance, the tyrosine derivatives with the sulfonyl fluorides.
Yeah, great question. Thank you very much for pointing that out. So my thinking is covalent small molecule drugs have been so successful And initially, people also were afraid of many off-target reactivities and idiosyncratic toxicities. The reason we started this program almost 10 years ago is we think proteins compared to small molecules, if you make a proteins into covalent version, it should have certain advantages over small molecules because, one, proteins have larger interaction interface, which can achieve better specificity, and proteins also have a large local microenvironment you can, ideally you can manipulate, but many times you could have a chance to, for example, blocking random interaction to trigger the reactivity. And another thing I want to point out, in our case, when the covalent, in order for the covalent bond to form, there are two steps involved. One is this protein A needs to bind with protein B, requirement 1. This requires some specificity. And requirement B is this unnatural amino acid side chain needs to point in the correct orientation and distance toward its targeted natural residue. Therefore, these two gates make this specificity, I think, is very high. In addition, I want to, we did some off-target reactions, both in mice and in human serum. And using Western blot, we haven't detected any off-target cross-linking yet. Of course, I understand that this is not sufficient. And for example, in our radio TRT experiment, we also monitored the secretion of the covalent antibody versus the wild-type antibody radio-labeled, and we didn't see any significant difference either, suggesting there's no significant retention or off-target reactivity either. But you have a great point. It's really important to address this, and I think as this covalent protein drug, this is at the very early stage, there's a lot of work to be done, and we think there's a great potential, but we need to be very careful to evaluate all these aspects as you suggested. Thank you.
Thank you.
Hi, Lei. That was beautiful. I have two questions. The first question actually sort of relates to the rate enhancement of the sulfonyl fluoride reaction. So have you done any sort of in vitro measurements that give you an idea of when you have a protein-protein binding followed by then reaction of the hydroxyl with the sulfonyl fluoride, what fold rate enhancement are you achieving compared to, let's say, just in solution reaction? And coupled to that, I have another question, which is that when you think about just the phenyl sulfonyl fluoride, and then that on a protein, are you actually retarding its reaction by essentially putting on a bulky substituent, which is the protein? So in other words, is the reaction as such on a non-engaged surface retarded because you essentially have steric inhibition by having a bulky protein? Can you comment on that?
Really interesting comments. The first one is how many fold of improvement we have this proximity effect. So when sulfix click chemistry was initially developed by Barry Sharpless, on the small molecule level, they actually need a catalyst to allow this reaction to occur. But later, further development shows that you can also have sulfix click chemistry without the catalyst. In our case, Barry and Jeff Kelly at Scripps, they did a beautiful experiments to test the reaction of this on the small molecule level. You can basically boil sulfonyl fluorosulfonate with aniline, and then boil many, many hours, and there's no reaction. I.e., if we let this reaction to occur, as you suggested, without any this proximity effect, they actually don't react. That's what they have multiple papers to demonstrate that. So I actually cannot measure an enhancement. But what we did is we measured the kinetics on the protein level. When we have two proteins binding together, we measured the K2 second rate constant of this reaction. And our result was in the range of 5 to 50 per mole per second. So this is not great, because the click chemistry are in the range of 1 to 10 to the fifth. And the Ross has a great reaction that he showed me today, which is reached to the highest level of click chemistry, 10 to the fifth. For us, the Sulfex currently is 5 to 50. But we recently found arginine can accelerate this greatly and improve. Maybe right now, I think we can reach to 500. Still far slower than the click chemistry top level. But I think this is quick enough for many reactions. And that's question one. And the question two, I found is very interesting. So you are thinking this reaction is stable in our protein context because protein are difficult to as a SN2 leaving group is difficult to leave because of you have this binding affinity.
Yeah, essentially, like if you have a bulky protein,
Right.
does
Bulky
that
group
essentially discourage reaction at the sulfonyl unless you have a protein-protein binding, because the protein acts as a, as essentially sort of, doesn't sort of encourage, that's a loose word.
This is related to Ken's question. So I think that's one of the advantages of using proteins as covalent. You can put a warhead into the proteins, and proteins have this bulky effect to prevent, or at least to minimize unwanted reactions with our molecules due to random interaction.
So I have another question, if that's okay. For your 2 nanobodies against the spike protein, there are, I think, a number of nanobodies. How did you select the nanobody? Did you choose the most tight-binding nanobody or the one that was just available at that time?
Oh, great question. Yeah, we actually didn't choose because at that time is during the pandemic. It's all in the mess. So we actually used whatever in the literature. The MMB6 was developed by Peter Wright group at UCSF, and the MB70 was developed by someone at Tsinghua University. So we didn't particularly pick which nanobody to use. Our strategy is, that speaks something of our strategy. It works so far in our hands, it works on any nanobody we've used it on. And many other proteins, because the tyrosine, lysine, and histidine is just everywhere on the protein binding interface. So we never failed so far to cross-link the targeted protein of interest using this Sulfex proximally enabled reactivity.
Thank you very much.
And these 2 nanobodies have actually 10-fold difference. One nanobody has 1 nanomolar binding affinity. The other one has 0.1 nanomolar. The MB70 has 0.1 nanomolar. Therefore, you see the improvement is 8 to 10. The other one has 1 nanomolar is like 40-fold.
Thank you.
Okay, we're at the top of the hour. There's a reception right outside. Please stay. One more time, thank the audience, the speaker.
Thank you.